viewers. Myself, C Sumathi. I am working as Assistant Professor, PG and Research Department of Biotechnology, JJ College of Arts and Science, Autonomous, Pudukkottai. Today we are going to see about enzymes used in recombinant DNA technology. These are the enzymes. Most widely used enzymes in recombinant DNA technology are discussed here. The enzymes are ligases, nucleases, restriction endonuclease, reverse transcriptase, DNA polymerase, ribonuclease, topoisomerase, alkaline phosphatase, polynucleotide kinase, terminal transferase, methylases, and gyrase. First, ligases. Ligases are the enzymes that join the nucleic acid molecules together. They can join both DNA and RNA molecules. If they join the DNA molecules, the enzyme is called as DNA ligase. If they join the RNA molecules, the enzyme is called as RNA ligase. DNA ligase is isolated from E. coli and bacteriophage commercially and used in recombinant DNA technology. The most widely DNA ligase is derived from T4 bacteriophage. The enzyme DNA ligase catalyzes the formation of a phosphodiester bond between the 5' phosphate of one strand and 3' hydroxyl group of another. It is used to insert the DNA fragments into the cloning vector. This diagram shows how the ligase enzyme join the two strands of DNA. It needs NAD plus or ATP for the energy the enzyme is needed. Next one is restriction enzymes. The word restrict means cut. The gene cloning requires the DNA molecules be cut in a very precise and reproducible fashion to insert the new DNA. First restriction enzyme was isolated in 1970. Restriction enzymes are also called as molecular scissors. Host control restriction. Some strains of bacteria degrade the foreign DNA by cleaving its DNA at specific sites by an enzyme before it takes time to replicate. The verb restrict means cut. Nucleases. Nucleases are enzymes that degrade DNA molecules by breaking the phosphodiester bonds. It usually cut DNA on either side of distortion caused by thymine dimers or intercalating agents. Nucleases that break RNA and DNA are called as RNases and DNases respectively. Nucleases are of two types. They are exonuclease and endonuclease. Exonuclease means it removes the terminal nucleotide of the DNA molecule. Endonuclease means it breaks the internal phosphodiester bond. These are most widely used ones. That is, endonuclease enzyme is most widely used in recombinant DNA technology. Then restriction endonucleases. The restriction endonuclease enzyme recognize and cut the DNA strand at specific sequence called restriction site. These enzyme is isolated from wide variety of microorganisms. Endonuclease enzyme degrades foreign genome when enter inside microbial cell, but the host cell own DNA is protected from its endonuclease by methylation of bases at the site of restriction. There are three types of restriction endonuclease: type 1, type 2 and type 3. The classification of restriction endonucleases. In this, type 1, type 2, type 3 are described briefly. Three subunit complex. Type 1 restriction endonucleases are three subunit complex. It has an individual recognition, endonuclease and methylase activities. It is ATP dependent enzyme. Cut both strands at a non-specific location. That is greater than 1000 base pairs away from the recognition site. Less commonly abundant than the type 2 restriction endonuclease. Example for the restriction endonuclease type 1 is ECO K1, ECO A1, then C4A1. Then type 2 restriction endonucleases. It is a homodimers endonuclease and methylase are separate In this enzyme, endonuclease and methylase are separate single subunit enzymes. It is a Mg2 plus dependent enzyme. It recognizes symmetric DNA sequences and cleave within the sequence. Most common about 93%. Example for this enzyme is EcoR1, BAMH1 and HIND3. Then type 3 restriction endonucleases. Endonucleases and methylase are separate two subunit complex with one subunit in common. This is the type 3 restriction endonucleases. It is a ATP dependent enzyme. Cleavage of one strand only. This enzyme cleave the one strand in a DNA molecule 24 to 26 base pair downstream of the 3' recognition site. It is very rare type of restriction endonucleases. Example for this enzyme is EcoP1, HINF3, then EcoP151. If the restriction enzyme cut the DNA strands, we can get the two type of products. First one is blunt end type, second one is sticky end type. If the blunt end means some restriction enzymes cut DNA at opposite base. They leave blunt-ended DNA fragments. These blunt-ended DNA fragments can be joined to any other DNA fragments with the blunt ends only. This diagram shows the restriction enzyme cuts opposite base. The two fragments we can get the two fragments in a blunt-ended fragments. Then second type of product is sticky end. Some restriction endonucleases cut DNA strands not exactly at the same position. Instead, the cleavage is staggered, usually by 2 or 4 nucleotides. So, the staggered cuts produce single-stranded overhangs at each ends of DNA fragments. These type of cutting is called as sticky end DNA molecules, or these are called sticky or cohesive ends. The next enzyme is reverse transcriptase, or simply called as RTase, is used to synthesize complementary strand, that is cDNA, from mRNA template. It is also known as RNA dependent DNA polymerase. The enzyme is used mainly for copying mRNA molecules in the preparation of cDNA for cloning. It is isolated from retrovirus, that is, reverse transcriptase enzyme is isolated from retrovirus. The next enzyme is DNA polymerase. DNA polymerase is a complex enzyme which synthesizes nucleotide complementary to template strand. It adds nucleotide to free 3' end and can help in elongation of strand. It also helps to fill gap in double-stranded DNA. DNA polymerase 1, isolated from E. coli, is commonly used in gene cloning. Taq polymerase, isolated from Thermus aquaticus, is used in PCR. The next enzyme is ribonuclease H, or RNase H, or RNH. Ribonuclease H is a family of non-sequence specific endonuclease enzymes that catalyze the removal of mRNA from DNA RNA heteroduplex. RNase H is also isolated from retrovirus. RNAH cleaves the phosphodiester bonds of RNA in a double-stranded RNA DNA hybrid, leaving a 3' hydroxyl and a 5' phosphate group on either end of the cut site. The types depend on the differences in their amino acid sequences. RNases H are classified into type 1 and type 2. RNase H1, that is type 1. These enzymes are involved in the processing of RNA primers from Okazaki fragments during DNA replication. And it is not essential in unicellular organisms. Then type 2, RNase H2. These plays a major role in ribonucleotide excision repair, that is removing misincorporated ribonucleotides from DNA. It is important for maintaining the genome stability. The next enzyme is topoisomerase. They regulate overwinding or underwinding of DNA. They are found in all types of cells, from man to all cells from to man. That is cells from to man. They also make incision in the DNA backbone. This enzyme make a incision in the DNA backbone. Classification based on the number of strands they break. The top it breaks the DNA strands. So it classified into type 1 and type 2. In type 1, this enzyme makes nick in one strand and possesses the intact strand through the nick and release the gap. And type 2, this enzyme makes double strand break and creates a gate through which a second segment of helix is passed. Mechanism of action of topoisomerase type 1 and type 2. This diagram shows in type 1, one strand is break by the enzyme. In type 2, both the strands are broken by the topoisomerase enzyme. Then some enzymes that modify the ends of DNA molecules that are discussed here. First one is alkaline phosphatase. The enzyme alkaline phosphatase helps in removal of terminal phosphate group from 5' end. It prevents self-annealing of vector DNA soon after cut open by restriction endonuclease. That is, it prevents recircularization of vector and increase the frequency of recombinant DNA molecule. The next enzyme is polynucleotide kinase. It adds the phosphate group from ATP molecule. The ATP molecule acts as a donor for phosphate group. Then donated phosphate group is transferred to a terminal 5' end of the DNA molecule after the dephosphorylation by alkaline phosphatase. The next enzyme is terminal transferase. It is an enzyme that converts blunt end of DNA fragments into a sticky end. We already seen that in endonuclease, there's a restriction endonuclease. There are two types of products we get from after restriction endonuclease action. That is, one is blunt end and second one is sticky end. In that, the blunt end molecule is blunt end DNA fragment is converted into a sticky end enzyme. Sticky end. This is catalyzed by terminal transferase enzyme. If the restriction enzyme cuts the DNA forming blunt ends, then the efficiency of ligation is very slow when compared to sticky ends. So the enzyme terminal transferase converts blunt end into sticky end. Terminal transferase enzyme synthesize short sequence of complementary nucleotides at free ends of DNA so that the blunt end is converted into a sticky end. The next enzyme is methylases. It is also known as methyl transferases. It is also called as methyl transferase. It was mostly found in bacteria and then bacteria to mammals. Usually, organisms that make restriction enzymes also synthesize DNA methyl transferase that protects their own DNA from cleavage. These enzymes recognize the same DNA sequence as the restriction enzyme they accompany. So methylation is the process of addition of methyl groups to adenine or cytosine base within the recognition site and thereby modifying the site and prevent the DNA restriction from the foreign restriction enzymes. Gyrase. For DNA gyrase is one of the types of topoisomerase enzymes called type 2 topoisomerase. We already discussed about the type 2 topoisomerase. That's all about the enzymes which are used in recombinant DNA technology. Thank you.